everyone, welcome to my garden. Today we're going to go over how to make a cast of the epidermal surface of different leaves. And we're going to use the example of two different varieties of cannabis. So what this type of procedure helps us do is it helps us see the shape of the epidermal cells on leaves or whatever surface we're making a cast of. And it helps us to count stomata on our leaves or other surfaces. Today we're going to answer the question, are there more stomata on the top or the bottom side of leaves on cannabis? And are there differences in number of stomata or cell shape between two varieties of cannabis? So let's get into it. This is our first plant that we're going to be looking at. This is a cherry lime reserve from Atlas Seed. Looking great. And I think what we're going to do is we're going to look at the oldest mature leaves on this plant. This is our second variety of cannabis, and this is a diesel sour Runtz hybrid from Atlas Seed. And this one, it doesn't have the same set of leaves, so we're actually going to pivot. We're going to look at this leaf, which is, since I trimmed it off, this is how many nodes up. We've got 1, 2, third node, so one of the leaves from the third node on each of these plants. For this procedure, we're going to need some clear nail polish, some clear packing tape. We also need our 2 leaf examples. We also need some blank microscope slides and a microscope. When we're doing these casts, we really only need a small part of the leaf, so I chose to isolate the middle leaflet out of the leaf, which is also the largest one. And then what I'm doing is I'm cutting that in half, both of them, because we're going to take a cast of the back and the front, and it's a little easier to handle when you have 2 halves of the leaves. So once I'm ready with those halves separated, I take my nail polish and just put a very thin layer over the interveinal space on one on the front and one on the back of each of my leaves, because we want to see the differences both between the front and the back and the 2 different varieties. While I'm waiting for the nail polish to dry, I'm going to go ahead and label my slides to keep everything straight. Once the nail polish is dried, we take a small piece of packing tape and gently press it to our nail polished section, and then we can kind of pick up the whole thing and gently press down to make sure that tape is getting some good contact with the nail polish. And then we can slowly peel it off, and you can see the nail polish comes right off. We want to be careful not to touch the tape too much so we don't have any fingerprints, and then we can put it right onto the slide to preserve that cast. We'll do this one more time on the other side as well, just to show you. Gently picking up the leaf with the tape, gently pressing down to get good contact with the tape on top of the nail polish, and then very gently peeling it back once we've gotten that tape all the way down. And the nail polish comes right up. And then we put that onto the next slide. Alright, so this is what I ended up with. Got our cherry lime reserve, our upper surface, and our lower surface. The lower surface was harder than I thought it was going to be. And then our diesel and sour Runtz, upper surface and lower surface. Let's take a look under the microscope. So this first view is at about 40x, where we can see kind of some bigger details, veins, and a lot of texture, but not a lot of detail. But we can see that our cast was successful. And at 100x, which is the next magnification up, we can really see cell wall details, and we start to see some of the finer details, like we can see a lot of trichomes here, and we can start to see stomata. But at 400x, we can really see the most amount of detail, where we can really start to see how those epidermal cells are arranged around trichomes. So here the trichomes would be the larger circular structures. And we can also see some details of stomata, which here it looks like we've caught a bunch of stomata in the process of emitting some gases. You can see some bubbles there coming out of them. So everywhere you see those two little bubbles, we have stomata. Very interesting. So all of these pictures were of the cherry lime reserve on the top side of the leaf. So this is the upper leaf surface on cherry lime reserve. And we'll take a look at some of the differences between the two varieties in our next post.